Okay, so now for the second part of the first part, okay, part 1.2, we need to calculate. We provide what we provide is a background of your gene expression for one specific sample, and we also provide you, where are the methylations, located, which we generated actually from the first question, right? So after the first question, we will be able to see all these methylation events through the entire chromosome, through the entire genome. And now we also have the background gene expression level. Now we want to see, what we want to check is actually, do we have a lower gene expression if we have a higher, more frequent methylation events? Because that's kind of the hypothesis here, right? If you have methylation, you know, through the chromatin regulation mechanism, you would have, in general, lower gene expression level. We also put here some, kind of positive controls, like wherever you have this histone modifier, those proteins, those enzymes, you would also be able to see some upregulated gene expression. So we ask here that, just trying to state the problem, computationally, what we are doing here is like we have a background gene expression and we have some location files. Now we want to look at the intersections. We want to see the overlaps of the methylation events and the gene expression and the genes, and we want to get the expression level of those genes. Okay, so the tool we are going to use is the STAT115. I'm actually not sure whether you have been working on this before in the previous homeworks, but it's fairly straightforward. You just load this tool, which is pre-installed already from Canon, and you call saying, I want to calculate the intersection between the A file and the B file. Okay, and the A file, you just put here the background gene expression data, and the B file would be the BAT file, which is the file that record all these locations of your events. Okay, and then we would ask you to write all those files into a new file. Basically, we want you to create a file and save all this output, and then later on you would need those output files directly. So here, just a very brief recap of the batch scripts. So using this vertical line symbol, is to basically is the bash language trying to append additional commands after you finish the first one. For example, let me see if I have some example here. Actually, we don't. Now imagine, say, I want to take a head of this file. Okay, imagine this is something like we want to do. Okay, so now basically what this cat or what this head is doing is just to pick the first of few lines, and the output of this function is a text stream. You have all this text. So next thing you can do is, now I can search for a gene of interest. For example, I'm specifically interested in this gene, and then I can just grab it. So what this is doing is you're taking the output of the original function, the previous function, and you feed into the second function, the second program. And the second program will take that as the input and gives you whatever you would need downstream. Okay, so other things can also work. It's like, imagine now I can cat the entire file and then do the head, which is exactly the same as what we did here. So just taking the head directly. Or I want to cat all the expression, all the files. I want to catch all the files, and I want to first search for, all the chromosome 1 genes. Okay, and that would gives you a lot, right? Okay, it also gives you 11. Let's say this would gives you no. Okay, let's try chromosome 3. Okay, so that would gives you only chromosome 3. Okay, so now you're interested in the first 100 genes. On chromosome 1 on chromosome 3 that we have from this expression. Then you put another vertical line. It's just like the the commands we use in R. You can just append whatever you need. So imagine here, now I want to see the head. That will gives you the first 10 chromosome 3 genes that we found from the file. Okay. The reason we are reviewing this is because we will need this for your homework. Because imagine this bad tool, similar to catch, would gives you a output stream. And you like there are two ways you can do it. One way is just you just print it out on your screen as what this cat is doing, right? And then you can copy paste manually from your command line window to a text file. But apparently that's not very efficient because especially now the files are generally very large. So the way we do that is to say we can take the head. Imagine this is what the output of the bad tools is. Okay, we do some bad tools, that intersect a file and -b file, and that would give you the string, the string output, that stream. And then now I want to say say calculate how many lines do we have. So that will be a word count, -l. So that will give you how many lines do we have from this. So, in a biological, from a biological perspective, that's answering the question that how many of the genes that we identified as overlapped with the methylation events. Okay. So that would gives you an idea of whether your analysis is making sense or not. Imagine that if that gives you a 0, that means you don't have any output. So the bad tool is not working properly. If that gives you a, I don't know, millions or billions, then that also not reasonable because we don't have that many genes. Okay, so this is like a sanity check. Another thing to keep in mind is that the bad tools would not automatically remove all these duplicates, because we would have many more methylation events, but we only have 20,000 genes in our genome. So there will be a lot of duplicates, let's say. And then what we need is actually only the unique samples from this library. So we can put, similarly as what we put before, like the head, we can put a unique here. This unique here would just delete all these duplicated lines from your file. And then you can count it again, saying, this asking how many unique genes we identified that has an overlapping region with the methylation event. And eventually, you want to save all these things into a file. Then you put this larger symbol, this greater symbol. That means I want to put whatever I got before this symbol into the file I specified after this symbol. So this so if you run this, you would get the intersection first, remove the unique, remove the duplicate, and save it into a TXT file. And you would need this for your later plotting. Okay. I see another question. Why are we doing the conversion again for part 1? Yeah, let me come back to that. Okay, after we finish this part 2, because it's slightly recorded. It's being recorded, so it's kind of slightly off. Okay, but I will come back to that. Don't worry. Okay, so that's the bad tools, the main function of the bad tools. Now after you do this intersection, you would have for each event list and each gene expression background file, you would have a result file generated with all this overlapping. And then what we are basically asking is, we have designed 4 groups of those events. Methylation is one type of event. We also look at for example, this H3K27 gene enzyme events. Okay, this modification, this histone modification events, and also the other types of methylation modification. So we want you to compare between this group, the methylation group, and the background, which is the general random background. Okay, so we want you to compare between these 4 groups to see if you see any consistent gene expression level change or not. Okay, so that's what we basically ask you to work on in part 1.2, which is actually it's a very routine kind of procedure if you want to work with this kind of data in the future. I think this this time the questions we put in the homework is very hands-on, and probably can also be very directly usable when you work on your own projects. Okay, any questions specifically for this sub question? Okay, if not, let me stop sharing this and let me stop recording this, and then we can come back to the question from Yao.